All right, if you're an IB biology student, you are in the right place. We're about to dive into the absolute foundation of the entire subject, the cell. This is Unit 1, and trust me, getting a solid grip on this stuff is going to make everything else that comes next so much easier.
So, let's just jump right in with a huge question. What are the fundamental rules that govern all life? I mean, what are the non-negotiable laws of biology that apply to everything from a tiny bacterium to a massive blue whale?
Well, the answer to that big question is actually pretty simple. It's something called the cell theory. And this isn't just a theory, it's really the bedrock, the core principle of all biology. You can think of it like a constitution for all living things, and it's built on three really profound ideas.
Okay, here they are, the three tenets. And they're elegantly simple, right? First, if it's alive, it's made of at least one cell. Simple as that. Second, the cell is the smallest possible thing that we can call alive. And third, you can't just magic a cell into existence. Every single cell has to come from another cell that already existed and divided. These three rules, they govern all life as we know it, period.
Now, this whole idea leads to a really cool concept called emergent properties. The best way to think about it is this. A single heart muscle cell can't do much on its own, right? It can't pump blood. But you get millions of them working together as a tissue, and suddenly they can create this coordinated contraction. That tissue then forms an organ, the heart, which can pump blood. That ability to pump, that is the emergent property. It's a new function that only appears when all the individual parts team up. The whole is literally greater than the sum of its parts.
So if the cell is our basic unit, does that mean they're all the same? Well, not quite. Life on our planet is essentially built on two fundamental blueprints. You've got the simple prokaryotic cell and then the much more complex eukaryotic cell. Let's break down how they compare.
Okay, you can see all the key differences laid out here, but if there's one word you need to remember for your exams, it's compartmentalization. Prokaryotes are simple. Their DNA just kind of floats around in a region we call the nucleoid. Eukaryotes, on the other hand, are masters of organization. They have a true nucleus to house their DNA, and they're jam-packed with all these little specialized compartments, or organelles, each doing a specific job. Oh, and see the ribosomes? Prokaryotes have the smaller 70S ones, while we eukaryotes have the larger 80S. That S just stands for Svedberg unit. It's basically a measure of size.
Now, every single cell, doesn't matter if it's prokaryotic or eukaryotic, has a gatekeeper. We call it the cell membrane, and it's this incredible dynamic barrier that is super strict about every single thing that gets in or out.
We describe this structure with something called the fluid mosaic model. It's mostly built from these amazing molecules called phospholipids. They have this brilliant two-faced design, a head that loves water and a tail that absolutely hates it. So when you drop them in a watery environment like inside a body, what do? They automatically arrange themselves into this perfect double layer, with all the water-hating tails hiding on the inside. We call it fluid because the parts can move around, and a mosaic because it's dotted with proteins and other molecules that do all the important work.
And speaking of that mosaic, a super important piece is the integral protein. What you're looking at here is a channel protein. Think of it like a secure tunnel that goes all the way through the membrane. It provides a safe, specific passageway for certain molecules that otherwise could never get across that oily barrier.
So how do things actually get across? A great way to think about it is like a river. Passive transport is just like floating downstream. It's effortless. Things just move from where there's a lot of them to where there's not so many, and it takes zero energy. But active transport, that's like trying to paddle upstream. You're fighting against the current, forcing molecules to go against their natural flow, and that takes a ton of energy, which the cell has to pay for using a special energy molecule called ATP. Okay, now osmosis. This is a super important type of passive transport that only, and I mean only, deals with water. Here's the key idea. Nature really doesn't like imbalance, so water will always move across a membrane to try and dilute whichever side is saltier or has more stuff dissolved in it. Just remember, water flows from an area with less dissolved stuff to an area with more dissolved stuff. We already know that all cells have to come from pre-existing cells, right? But how does that actually work? That question brings us to the life story of a cell, which we call the cell cycle. And this cycle culminates in the absolutely incredible process of mitosis. So a cell's life isn't random, it's highly structured. It actually spends most of its time in the G1 phase just being a cell, doing its job, growing bigger. Then when it gets the signal to divide, it enters the S phase, S for synthesis,
where it carefully copies every last bit of its DNA. G2 is like the final prep stage, checking to make sure everything is ready. And then, it's time for the main event, the M phase, or mitosis, where the cell actually divides in two.
But how does a cell know when to move from G1 to S or from S to G2? Well, the answer is a group of proteins called cyclins. You can think of them as the traffic cops of the cell cycle. Their levels go up and down, and once they hit a certain concentration, they basically give the go-ahead signal, pushing the cell past a checkpoint and into the next phase. They make sure everything happens in the right order at the right time.
And now for mitosis itself. It's like this beautifully choreographed dance. In prophase, the DNA coils up super tight so we can see it. In metaphase, all the chromosomes line up right at the equator. Just remember, M for middle. In anaphase, the identical copies get pulled apart to opposite sides, A for away. And finally, in telophase, two brand new nuclei form, and you end up with two perfectly identical daughter cells.
So what happens when this incredibly regulated cycle goes haywire? Well, that's what cancer is. At its very core, cancer is a disease of the cell cycle. It's what happens when mutations break the genes that control those cyclin checkpoints, essentially telling the cell to ignore the stop signs and just divide and divide and divide without control.
So why does all this fundamental knowledge matter so much? Because once we understand these basic rules of life, we can actually start to use them. This understanding is opening up absolutely incredible possibilities in medicine. And there is no better example of that than the exciting field of stem cells.
Stem cells are basically the body's master cells. They're undifferentiated, which means they're like a blank slate. They haven't decided what they want to be yet, and they have this amazing potential to turn into any type of specialized cell. A nerve cell, a muscle cell, a skin cell, you name it.
And this isn't just some science fiction idea, this is really happening now. For a condition called Stargardt's disease, which causes blindness, doctors can take embryonic stem cells, coax them into becoming retinal cells, and inject them to actually restore a person's vision. Or for leukemia, doctors can use stem cells to completely rebuild a patient's blood and immune system after it's been wiped out by chemotherapy. It's truly revolutionary stuff.
So we've gone from the most basic rules of the cell all the way to the cutting edge of medicine. We're not just learning to understand cells anymore, we're learning how to guide them. And that leaves us with a pretty profound question to think about. If we can truly direct cells to become whatever our bodies need them to be, are there really any limits to what medicine can achieve in the future?